Why colony strength and the right bees matter for wintering. Winter survival of honeybee colonies depends a lot on how many bees were raised at the end of summer and on the overall strength of the colony. Strong colonies with a large share of young bees tolerate winter conditions well and build up quickly in spring. Weak colonies with too few young bees usually get even weaker during winter and can take a long time to recover the next spring. Sometimes, even colonies that look strong in autumn can have a lot of dead bees during winter and become weak by spring. This happens when mostly older bees go into winter, bees that did not rest after the main honey flow. These bees often do not survive until spring and die during winter, creating a large amount of dead bees in the hive. Colonies winter best and then raise a lot of brood in spring when many of the winter bees were born in late summer, end of August and in September. These bees are physiologically young because they did not spend the autumn raising brood. Special trials with colonies made up of different proportions of young and old bees showed that colonies winter better when young bees dominate. In one trial, two groups of colonies were formed with the same total mass or size. In the first group, most bees were older, born in midsummer. In the second group, most bees were younger, born in late summer. All colonies wintered indoors in a cool wintering room at about 37 to 45 degrees Fahrenheit, 3 to 7 degrees Celsius. Conclusion. One important condition for successful wintering is having many young bees born at the end of summer. Colony strength is the second key condition. The second important condition for good wintering is the strength of the colony. To keep the best internal microclimate during winter, bees must work and spend energy. The weaker the colony, the more energy and food the bees spend in winter to maintain the necessary temperature inside the winter cluster. As a result, bees wear out more during winter and become even weaker by spring. Strong colonies use much less honey per unit of colony weight than weak colonies. Because of this, bees from strong colonies are less exhausted by spring. The stronger the colony going into winter, the less feed it consumes per unit of mass. A research institute conducted an experiment that clearly showed the relationship between colony strength and the amount of feed consumed. In autumn, colonies were formed with strengths from about 4 to 5 to 14 to 16 bee-covered frame spaces, seams, and they wintered in a semi-underground wintering room at about 32 to 36 degrees Fahrenheit, 0 to 2 degrees Celsius, very cold conditions. The lowest winter feed use was seen in colonies of about 9 to 12 seams. The highest feed use was in the weakest colonies, about 4 to 5 seams. It was also found that very strong colonies, 14 to 16 seams, consumed a little more feed. This is explained by the fact that with many bees, the cluster temperature stays higher in winter. So the best optimal colony strength depends on wintering conditions. in harsher, cooler conditions, and especially wintering outdoors, stronger colonies are needed. Strong colonies have other winter advantages. Strong colonies not only consume less honey per unit of mass, but they also do better on other wintering indicators. In strong colonies with many young bees, there is much less dead bee accumulation in spring, and the nests look cleaner. Bees in strong colonies are less affected by nosema and other diseases, and they raise more spring brood. Because of this, colonies that winter well grow rapidly in spring and collect a larger amount of honey in summer. How beekeepers build strong colonies with many young bees. Several methods have been developed to increase the raising of bees at the end of summer and form strong winter colonies with a large share of young bees. These methods are widely used in well-managed commercial apiaries, and as a result, wintering success improves. Beekeepers also noticed that young queens, this year's queens, lay more eggs at the end of summer, and brood remains present longer in their colonies. As a result, colonies with young queens go into winter stronger and with more young bees. One researcher, mid-1900s, reported that in colonies with young queens, there was more brood in autumn than in colonies with older queens. To stimulate egg laying during this time, bees need at least a small supporting flow. This can be achieved in different ways. For example, colonies can be moved to a late-blooming nectar flow, or nectar plants can be planted so they bloom in autumn. In some regions, many colonies are moved near the end of summer to an area with strong late summer flowering. By collecting nectar and pollen there, colonies increase brood rearing, become stronger, and later winter better. Compared to colonies that stayed in place, these colonies have more young bees and winter better. Even a small daily intake of nectar, about 200 to 300 grams per day, about 7.1 to 10.6 ounces, or 0.44 to 0.66 pounds per day, when young queens are present, can significantly increase brood rearing at the end of summer. If there is no nectar flow at all, egg laying can be stimulated by feeding colonies sugar syrup at about 200 to 300 grams per day, again 7.1 to 10.6 ounces, or 0.44 to 0.66 pounds daily. A supporting late flow or feeding small portions of syrup increases the amount of brood in colonies by about 30 to 50%. Protein, pollen or bee bread matters too. To increase brood in colonies, besides nectar or syrup, it is also necessary to have enough protein feed, meaning bee bread, stored fermented pollen. If colonies have little bee bread and there is no pollen flow in nature, the bees should be given additional protein feed. Research showed that autumn protein feeding has a positive effect on preparing bees for winter and on wintering. Colonies that received protein feed raised 10 to 11% more bees than control colonies that received only sugar syrup. They also wintered better, 23.8% less dead bee accumulation, 8.7% less feed consumed over winter, 21% less nosema infection in spring.